Hello, General Biology, and welcome to the third in the screencast series for Chapter 7, Sections 7.3 and 7.4. And we just got done the very last tail end bit of 7.2 with the cell membrane. Hopefully you're comfortable with that because now when we talk about cell transport, we're going to have to use what we learned from that cell membrane, and that's one of the reasons why I wanted to put these together. So what we're going to do right now is we're going to watch one of my favorite little YouTube videos from the Amoeba sisters. So hold on and let's watch the video.
What it might be like to be inside a cell. Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose. But what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates. And when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, that's a great introduction to what we're going to be talking about in this screencast and in a couple other screencasts as well. So let's talk about molecules. And molecules, no matter whether they're in a liquid, a solid, or a gas, they are in constant motion. Gas molecules move very, very fast. They bump and they collide with each other and they bounce off of each other. Liquid molecules, they move pretty fast as well, and we can see that through the process of evaporation. They bounce off of each other as well, but they're not quite as fast as gas molecules. And then solids, and you're going to say, no, Mrs. Saunders, something that's a solid doesn't move. It does. It just moves so slow that with our eyes we can't detect it. But if we were to have a really strong microscope, we would be able to pick up that motion. We would be able to detect it. So this movement of molecules being in constant motion is called Brownian motion. And of course it was named after a man by the name of Brown. So what we know about this is, and I liked this picture and this is why I picked it out. If we had a beaker of water and we drop some food coloring in, what's going to happen is that this liquid is already moving. The food coloring molecules are going to disperse. They're going to start to move apart until they get evenly distributed throughout that beaker. And this movement from an area of high concentration right here to an area of low concentration, which this is the low area, this is the low area, is referred to as diffusion. So diffusion, again, let me define that for you. It's written here, is the movement of molecules from an area of high concentration to an area of low concentration. It doesn't require any energy because the molecules move with the flow, and the flow is always going to be from high to low. So by adding this food coloring, we've created a high concentration of food coloring here, and there's a low concentration of food coloring here. So the molecules are going to move from here to here, and as I said, until they become evenly distributed. And when they become evenly distributed, that's called equilibrium. Now the molecules don't stop moving, they're still moving, but we can't see the overall change because they're equal, but it is moving. Now that area from high to low, we're going to refer to that as the concentration gradient. So when the movement goes with the gradient, it is going from that high to the low. Now remember, inside of our cell membranes, we have proteins. So here's a cell membrane. These are the proteins. Some of these proteins can form channels, and some of these proteins can form like a gate. Now, I remember my sons, they had a sandbox when they were growing up, and I had this really cool toy that had like this water wheel on it. And when you put sand in it, it caused the wheel to spin. And the sand would go to where there was a trap door. And as soon as you got enough weight from the sand, that trap door would open and it would drop that sand out. And that's basically how this gate protein works. It'll open up because there's molecules being pulled in with the natural flow of diffusion. So here's an example with this. Here's molecules that we have a greater concentration. Remember when we looked, and I'm going to flip back here. Remember when we looked here and we saw that these are the heads and there's openings in these heads? Same things happening here. We just can't see it from that angle. So because there's a higher concentration and because the heads are pulling water in, they're just going to go with the flow. Almost like if you've got a boogie board and you're out in the ocean and you're just riding that wave in. So here is a transport protein that's a channel protein. Again, we've got a high concentration of molecules here. They're not going to be able to fit as easily through the spaces, so they're going to move through the flow with this channel, and they're going to pass through it. And we talked about this being a gate. This gate is going to open up. It's going to allow a few to come in, and they're going to be able to go to the other side. So this is a form of passive transport, and that's still where the movement of molecules are moving through the natural forces of diffusion from a high concentration to a low concentration. And specifically, this example of passive transport is called facilitated diffusion. And this is, as I said, a form of passive transport that utilizes proteins to move the molecules against the gradient. Well, we're done another screencast, so I will see you in just a few minutes for the fourth one.